Rhodopsin. What is rhodopsin? Rhodopsin is a pigment containing sensory protein that converts light into an electrical signal. Rhodopsin, or also called phototransduction, is classified as a chromoprotein where from its name chromo means colors. It's made up of a colorless protein called opsin and a pigmented molecule derived from vitamin A which is called 11-cis-retinol. Have you ever wondered how rhodopsin works? Well, the back of our eyes in the layer of tissue known as our retina, we have two kinds of photoreceptors cells called rods and cones. While rods help you see in dim light, cones function best in bright light. Both kinds of cells contain photoreceptors that signal to brain in response to light, but rhodopsin is the one found in rod cells. In fact, rhodopsin was the first G-protein coupled receptor or GPCR to have its crystal structure determined by X-ray crystallography. When rhodopsin absorbs light, it undergoes a conformational change triggering a signal transduction cascade that sends a signal to the brain that gets interpreted as vision. So how exactly does rhodopsin do that? Retinol is a ligand composed of a proline chain and a beta-ionone group. When rhodopsin is active, retinols bind to opsin, but upon photoactivation, the ligand dissociates. The opsin core is made up of 7 transmembrane alpha helix. It has 3 loops that extend to the extracellular matrix including the N-terminus and 3 loops that extend into the cytoplasm including the C-terminus. The transmembrane region contains the most hydrophobic residue shown in red, while the extracellular and the cytoplasmic loop contain more hydrophobic residue shown in blue. The hydrophobic core allows the protein to sit within the nonpolar membrane of rod cells. The second loop on the extracellular side of the protein is particularly important for the binding retinol. This region folds into 2 beta sheets stabilized by disulfide bond. The beta sheets serve as a lead blocking retinol from dissociating when rhodopsin is inactive. Another important feature is the salt bridge between arginine 135 and the glutamate 247, which prevents G protein from binding to inactive rhodopsin. Now that we understand the structure of rhodopsin, let's talk about how it functions. Under light conditions, rhodopsin sends visual cues for a process called phototransduction. When a photon hits 11-cis-retinal ligand and rhodopsin, it isomerizes into 11-trans-retinal. The isomerization of a retinal triggers a conformational change in the opsin protein, and through a series of intermediates, it turns into meta-rhodopsin 2, the active form of rhodopsin. At this point, the shift-based linkage between the 11-trans-retinal and the lysine residue is hydrolyzed, allowing 11-trans-retinal to dissociate from the complex. The G protein active site undergoes various changes as well. Helix 6 tilts away from the transmembrane cores at the cytoplasmic side, and this results in the widening of the binding site. Rhodopsin, being a G-protein coupled receptor, will bind to the alpha subunit of the G-protein translucent when photoactivated, and it's stabilized by the hydrophobic residue cysteine 140. The binding results in the conversion of a GDP to GTP, and the GTP bond alpha subunit then dissociates from the beta-gamma rhodopsin to activate cyclic GMP, phosphodiesterase or PDE. Phosphodiesterase then hydrolyzes cyclic GMP, and this decrease in cyclic GMP causes sodium and calcium channels to close, leading to the hyperpolarization of the subsequent release of neurotransmitters. The signal then passes through several cells before reaching the brain to be interpreted as vision. The signal also tells the brain that light is present, leading to the switch from rods to cones. This process is called bleaching. Thank you for your attention.